Hello, my name is Bamzai-Wokhlu, and today I'm going to present a microarray analysis study of apoptotic pathways in SARS-CoV-1 versus SARS-CoV-1 delta E. COVID-19's deadly precursor, SARS-CoV-1, was responsible for the SARS epidemic in the early 2000s. Studying the mechanisms of this virus may be of relevance to the current COVID-19 outbreak. The pathogenicity of SARS-CoV-1 in particular has been shown to be highly attenuated with the missing envelope gene. Therefore, we reanalyzed open-source microarray data available from a cellular model which compared the stress and inflammatory responses of wild-type SARS-CoV-1 to SARS-CoV-1 delta E lacking the envelope gene. Our research uniquely analyzed the 24-hour time point, which may be critical in early stress response. So we used the GSE dataset GSE 3589 from the NCBI, and it contained microarray data from SARS-CoV-1 and SARS-CoV-1 delta E infections of endothelial cells ex vivo at varying time points. Using GO2R, we examined the differences in gene expression between endothelial cells infected with SARS-CoV-1 delta E and those infected with the wild-type SARS-CoV. We then used functional enrichment analysis in StringDB to identify enriched KEGG pathways, with the gene upregulation being defined as log FC values of greater than 0, and we used an alpha level of 0.01, so we only considered p-values less than that. We also calculated the false discovery rate of the TNF pathways and IL-17 pathways to be 0.0019. From there, we use gene cards and gene ontology to determine commonalities in the gene function of the genes that were common to both the IL-17 and TNF signaling pathways. So we found June FOS, TNF AIP3, NFK BIA, June B, CEPB, SOC S3, and to be common to both TNF and IL-17 signaling pathways. And here is their log FC value and their p-value. We further explored the functionality of these 7 genes using gene cards and PubMed to identify their roles within apoptotic and necrotic cell death pathways. Some of our conclusions included that the absence of the envelope gene expression in SARS-CoV-1 delta E resulted in an upregulation of cell death pathways. The TNF pathway regulates necrosis or cell death from external stimuli, whereas IL-17 helps regulate apoptosis. We also hypothesized that the envelope protein may have a role in suppressing or downregulating transcriptional factors like TNF AIP3 that otherwise promote cell death. By suppressing apoptotic pathways, the wild-type virus may sustain in its host. However, it should be remembered that these pathways are highly complex. All right, thank you so much. We want to thank the Buchholz Guidance Club